The overall goal of this procedure is to quantify the impact and end fate of heavy metals introduced into microalgae growth systems as a result of carbon dioxide from coal power plants being used as the carbon source for growth. This is accomplished by first understanding the potential contamination levels based on integrating coal flue gas with microalgae cultivation. These levels are used as baseline contamination and stocks are made. Next, the required inoculum is cultivated and the large-scale growth platform is set up. Then, the system is inoculated and samples are periodically taken for processing. Finally, the biomass is isolated and the end fate of the contaminants is quantified. Ultimately, inductively coupled plasma mass spectrometry, or ICP-MS, is used to determine the metals that are absorbed by the biomass, and combined with optical density measurements, enables the impact of the contaminants on the system to be understood.
The main advantage of this technique over existing methods is the ability to directly quantify the end fate of a multi-metal system. Though this method can provide insight into the impact of heavy metals from flue gas, it can also be applied to other systems.
This method can help answer key questions in the microalgae field, such as the impact of heavy metals on productivity and the ability of microalgae to be used for bioremediation.
Generally, individuals new to this method will struggle because of the potential for contamination, the required precision in preparing samples, and analysis with an IC.